The human body is an extremely complex system of many cells and organs working to stay alive. When a foreign invader enters the body, it has a sophisticated system to fight it off. However, in the world of excessive antibiotic usage and powerful bacteria, the immune system is not always successful. Today, I will give you a little look at the war that's waging between antibiotic-resistant bacteria and the human body. Bacterial cells attempting to invade the body are MRSA, tuberculosis, and Clostridium difficile. MRSA, also known as methicillin-resistant Staphylococcus aureus, is a type of staph infection, mainly dangerous because of its strong resistance to all antibiotics in the beta-lactam family. It is much more difficult to treat than a common staph infection, and as a result, it causes 11,000 deaths per year in the United States. Clostridium difficile is a bacteria that causes potentially lethal diarrhea. These infections usually happen in people who are recently exposed to medical care and antibiotics. Infections of Clostridium difficile are very aggressive and cause roughly 14,000 deaths a year in the United States. Drug-resistant tuberculosis is one of the most common infectious diseases and causes a large number of deaths worldwide. Usually airborne, the disease often affects the lungs. Tuberculosis that is resistant to drugs is harder and more expensive to treat, and in 2011 it caused 1,042 deaths in the United States. The immune system has a strong defense mounted to prevent foreign objects from entering the body. The skin is an excellent barrier. Unless there is already some kind of opening in the skin, such as a cut or scrape, it is an impenetrable wall. However, as it may be known, there are several key openings in the body where it is very easy for bacteria to enter, like the mouth, nose, and eyes. Each of these areas have special defenses in place as well. Mucus in the nose and lungs trap bacteria and push it upwards in an attempt to get it out of the body. Saliva contains acid that kills some bacteria. If the bacteria is able to get into the stomach also contains acid that is able to kill bacteria. Tears flush out bacteria that get into the eyes. If bacteria does happen to enter the body, the immune system immediately gets to work fighting it. Macrophages are a type of cell that devour bacteria and other foreign invaders. When the macrophages ingest the bacteria, they allow the other cells in the body to see antigens of the recently digested bacteria. These antigens are extremely important because they allow the immune system to see what is a foreign cell and what is a body cell. A T cell, which comes from the thymus gland, a gland located in the chest, reads the antigens and signals the immune system to respond if it is a foreign substance. B cells, which are produced in the bone marrow, read the antigen as well. But if they recognize it as a foreign substance, they will produce antibodies. Antibodies are proteins that are able to bind to the antigen and prevent the bacteria from carrying out chemical reactions. These antibodies also work as signaling flags. If many antibodies attach to one bacteria, it signals to the macrophages to kill it. Despite all the effort and energy the body uses to protect itself from bacteria, it is not always successful. That is when people look to antibiotics to finish the job. It is important to note that bacteria are only useful when fighting a bacterial infection or disease, because they only work against living organisms. Antibiotics are able to stop bacteria from multiplying or growing, but it cannot do the same for viruses because viruses are not living. A virus is essentially a single strand of DNA protected by a protein capsule. With this said, we can now look further into the function of antibiotics. There are many ways that antibiotics can be classified, but one way is to look at how they affect a bacterial wall. Bactericidal antibiotics kill bacteria by preventing the total formation of a bacteria's cell wall, and this causes the bacterial cellular contents to leak. Bacteriostatic antibiotics prevent the bacteria cell from continuing to multiply. Antibiotics are heavy duty and have a damaging effect on most bacteria they encounter. So how do humans remain virtually unharmed by taking these drugs? Most bacteria cells have a cell wall that contains the molecule peptidoglycan, which is a collection of amino acids. Human cells do not contain peptidoglycan, so the antibiotics do not harm the human cells. Antibiotics have only been used for a small period of time in human history, and some people already believe that these antibiotics are doing more harm than good. The use of antibiotics is causing bacteria to become stronger and more resistant to common drugs, making them much harder to treat. Bacteria become resistant to antibiotics in a wide variety of ways, and while excessive use of antibiotics should not necessarily cause it, it causes the process to progress much faster. Genes located on the plasmids that code for resistance are one of the most common ways resistance occurs. The author of the antibiotic paradox, Stuart Levy, says, quote, "Plasmids are small pieces of self-replicating double-stranded DNA that are accessory genetic elements in the cell. There may be as few as one or as many as 1,000 copies in a cell. They provide other traits not specified by the cell's single chromosome." Unquote. Plasmids are not part of an original cell's DNA, but are passed to cells through other cells. Transposons, similar to plasmids, are small pieces of DNA, but unlike plasmids, they can move to a different host if their current host dies. Plasmids and transposons play such essential roles in the progression of antibiotic-resistant bacteria because they are supplementable pieces of DNA that can potentially help the cell survive different situations. An example would be a bacteria cell like MRSA receiving a plasmid that has a gene that codes for penicillin resistance. If the bacteria comes into contact with the penicillin, it will survive, but the cells that do not have the plasmid will die. The reduced number of cells mean that the bacteria that has resistance to penicillin has much less competition for food and can reproduce more quickly. If an antibiotic is not used properly, the effects it can have on an individual, as well as the general population, can be quite dramatic, effectively increasing antibiotic resistance. If a person takes antibiotics for a shorter time than instructed, the weaker bacteria cells will be killed off, but the stronger cells will remain. Without any higher doses of antibiotics to kill off these stronger cells, they can grow and flourish with very little competition, resulting in a strong strain of bacteria. This is all very scientific. We have a general idea of how bacteria becomes resistant to antibiotics, but what does this actually look like? Most resistance is either altering the proteins that the antibiotic targets or using enzymes to prevent the antibiotic from functioning. For example, bacteria fight the antibiotic family of aminoglycides by coating the antibiotic with a chemical residue that makes it impossible to enter the cell. This would be an example of how using enzymes to prevent the antibiotic from functioning. The other main way bacteria show resistance is by changing the protein that the antibiotic targets. With no recognizable proteins to target, the antibiotic is useless. If antibiotics do happen to enter the cell, some bacteria have efflux pumps, which are a mechanism that pump out the antibiotics faster than they can enter the cell. People approach antibiotic resistance with several different viewpoints. One common belief is that the development of new antibiotics will continue to outpace the development of resistance to bacteria. This is not the case. No new classes of antibiotics have been discovered for quite some time. With more money to be made from drugs that treat chronic illnesses, most pharmaceutical companies have abandoned the idea of creating antibiotics that are only used for a period of days or weeks. Bacteria will eventually develop resistance to most or all antibiotics, and any new substances we introduce will only temporarily decrease the speed of resistance. While the process of developing resistance is entirely scientific, most of what can be done to change the outcome is possible by us. By becoming more responsible for more of our actions, we can change the scary scenario: a world without antibiotics.